Right, this is the field investigations required practical. Right, and what we're doing here is we're measuring the population size in a habitat using sampling techniques, and there is two. Number one is using a quadrat, number two is using a transect. Right, so firstly, what a quadrat is, it looks a bit like this. Right, it's usually 1/2 metre by 1/2 metre, so 0.5 metres or 50 centimetres, right, on each side. And what you do then is you get your field. So here's your field, right, just circled it in purple there, there's your field. And the idea then is you've got to estimate the number of daisies, dandelions, sunflowers, rubbish, whatever it may well be in a field. Now the best way of doing it is to make things random, right? We have to be talking about random here, or else what people will do is they'll just get the quadrat and they'll just aim for the place where there's the most daisies, right? What it does, it sort of disrupts the data. So possibly the best way of doing it is to get your field and turn it into a grid. Then you've got two numbers here, right, which are collect two numbers, one from each bag, and make it random. So you've got one bag and you've got another bag, and each side of each of those bags there is 6 numbers. Pull out a number, let's say you pull out 2. Then for the second number we pull out 3. Right, then what you do is you go up to 2, 3, and you are then, let's do it in black, you're then going to do that quadrat there, that little square there, right, and count the number of daisies or whatever it may well be. So you lay quadrat on the ground. Once you've done that one, what you then do is you put the numbers back in and you repeat it. You repeat it and then you count the number of your chosen species inside the quadrat again. And then you repeat it again and repeat it approximately 10 times. When you've done it 10 times, you then calculate your mean. Alright, so let's say your mean worked out to be 4 daisies per one, right? It's never going to work out probably as 4, right? What you then need to do is then you need to do a calculation that is going to work out an estimate for the number of daisies on the actual field. I've put a little equation here. So the estimated population size is the total area sampled times by the mean for each quadrat. So what I could do is I could do this here. So the total area sampled, right, is 6 metres by 6 metres. So that's 36 metres. But because a quadrat is then only 1/2 metre, I've then got to times 36 times by 4, which comes out at 144 of the quadrats. Okay. If my mean per quadrat was 4, right, in each one of these individual quadrats here, and I've done it and I've got the mean, I would then do 144 times by 4, which will then give me the total number of daisies in that field. The second example is something called a transect. Now again, you kind of use the quadrat device, right, but it's not quite the same. What you're looking now is you're looking for patterns. So rather than the total number in a field, you're looking for the pattern from one place to another. So on this one, what we could do is we could be starting off with our tree, right? And what we do is we put our transect, that is a 30-metre piece of tape measure, in a line from the base of a tree to an open area of ground. So it starts there and it goes 30 metres all the way to the end. So that's 30 metres. Then you put the quadrat against the transect line. So here's my first quadrat. I put it there, right? My transect goes out from the tree randomly. That's my first quadrat. I count the number of plants inside that quadrat that I'm interested in. I also then have to use a light meter to measure the light intensity at each position. So here I've counted and I've done light intensity. Record it in a table. Then every 5 metres, what you then do is you put your quadrat down again. 5 metres later, you put your quadrat down again. And you do exactly the same. Each time what you do is you count and do the light intensity, count and do the light intensity, right until you get to then 30 metres away. What you then do is then you will use your data, right, and put it into a results table that looks a bit like this. Now I don't quite know why it's come out blurred, right, but what I've got is that is directly underneath the tree. That's the number of lights or plants in the quadrat. That is then the light intensity. You then need to present your data in some way. Now, this at the top is kind of a little bit practical, where you're starting underneath a tree, you're going across a field, and you're ending up in woodland. Right? You could forget about the woodland. You just got to look at exactly what the examiner's talking about. You could just start underneath a tree, and you can end at the end of a field or in a car park or whatever it may well be. A good way of doing it is just to show it as a bar chart, where you start off, number of dandelion plants, underneath the tree there's none, then there's 2, and then what happens is as you go across the field, the number increases. 16 metres away, there's obviously an issue, maybe there's a bald patch of ground or whatever it may well be. And then what you do is you notice as it goes further along, it decreases again. Another example, right, could be a transect going from marsh across a bit of water and into a marsh again. And so what you can then do is you can then look at the different plants that you will then see starting from here, right, going all the way to here. Right, or there in fact, and all the way to there. And it's just another way of actually presenting a transect. Okay, so the 2 different methods here. You've got a quadrat, right? Quadrat originally is used to kind of estimate the number of something in a field. Daisies, rubbish, buttercups, whatever it may well be. Okay, but the biggest and most important part of that is random. The second one is a transect, and what a transect is doing is looking for a pattern, right? So you're not looking at doing any means or anything like that, you're looking for a pattern, right, that you can then explain based on your kind of biological knowledge. So for example, if you start under a tree, there's no light, or very little light, so the dandelions aren't going to grow. In the middle of the field, there's plenty of light. Also underneath the tree, there won't be as much water. Right, so then you've got to think about your abiotic and biotic factors.